Hello and a very warm welcome to the Rylands Lunchtime seminar series. My name is Stefan Hans from the John Rylands Research Institute and Library at the University of Manchester, and I'll be chairing this session today. The Rylands Lunchtime seminar series highlights the breadth and quality of our unique special collections and showcases world-class collections-based research activities across the university. We are running today's session remotely from a number of locations in and beyond the University of Manchester, as well as from various rooms in the historic John Rylands Library. And as usual, we will be showing some of the outstanding artifacts of our collections and presenting new research. Our seminar today is entitled DNA in the Archive, Using Genetics to Trace Manuscripts and Books. But before I introduce our speakers, there are just a few things to mention. This event is being recorded. It will be edited and made available on the John Rylands Library YouTube channel. Auto subtitles are available using your own settings, and we are using Zoom webinar format. So this means that your camera and mic is disabled on entry. And at the end of the session, there will be around 15 minutes for questions. So if you would like to ask one, and I highly encourage you to do so, please can you add questions to the Q&A function. This will be then selected by me as the chair and conveyed to the panel. We should have time to take a few questions, and we are aiming to stop around 1:45 p.m.-ish. So without further preamble, I would like to introduce our speakers today. Jerome de Groot is Professor of Literature and Culture at the University of Manchester. He writes about public history, genetics, and historical fiction. He has written Consuming History in first edition in 2008. A second edition was published then in 2016. And the volume, as I just heard, is just about to go into a third edition now. Jerome published various other books, including the historical novel, 2009, and he began to write about family history and genetics in 2018. And his book, Double Helix History, came out in 2022, a book I really enjoyed reading. Double Helix History is really a field-shaping publication that examines the interface between genetics and history in order to investigate the plausibility of new knowledge derived from scientific methods, and it reflects upon what it might mean for the practice of history. His project on DNA and identity, Manchester's DNA, entitled, has been running for some time now and brings creative practitioners into dialogue with genetic science. He joins us today from the Swedish Collegium for Advanced Study in Uppsala. And presenting with Jerome is Jeremy Penner, curator of African and Near Eastern manuscripts at the John Rylands Research Institute and Library. He holds a PhD in religious studies, and he has published widely on the ancient Mediterranean world, for instance, Patterns of Daily Prayer in Second Temple Period Judaism, which was published with Brill in 2012. Thank you both for talking to us today, and over to you.
Thank you, Stefan. I kind of revealed myself. I felt like I should have had my camera on earlier. Sorry. Hi, everyone. My name is Jerome. As Stefan says, I'm in Sweden at present. I'm super excited to share this information and research with you. This is stuff that is really cutting edge and new for me. I've been writing this over the last couple of months. So I'm interested to see what you think about it. It's also relatively raw. And I'm kind of aware that it's that some of the implications and some of the thoughts that I have might need attending to. So it'd be really fascinating to see your thoughts from all kinds of perspectives and your ideas and your questions. So please keep those coming in, because yeah, as I say, this is kind of work that I think it has many really fascinating implications for the way that we know things and for what we do within a library setting particularly. And I'm really hopeful that we can kind of explore some of those ideas together. What's going to happen is that I'm going to talk for about 10 minutes or so about these new kind of approaches to thinking about library collections and archives. I'm then going to hand over to Jeremy. He's going to show you this very beautiful piece of work that we're going to use as a kind of talking point and he'll talk a little bit about that piece. Then I'll say a few things about what the implications of that piece might be for us, given what I've talked about to begin with, and then we will. Then I'm going to talk a little bit about the future. And that's super strange. So we shall see how we go with that. I'm going to share some slides. I hope you can see them all. And I'm just going to talk through them. I'm afraid I'm a relatively undynamic presenter at this point. As I say, keep on coming in with questions and we'll try and get to them all. But I'm going to share my slides with you now, and I hope that you can see all my slides. Can I have a thumbs up from someone in my room just to make sure that they're all going okay? Yay, there we go. Fabulous. Excellent news. Thank you both. Okay, so yeah, this was originally entitled DNA in a library setting, but actually I've started really thinking rather more about the biomolecularization of the archive, and that hopefully I will explain what I mean by that. And what I'm going to do is essentially explain this as a sort of concept, as a means for starting to think about our collections and ways that we might start to approach them, or have been approaching them over the last 5 or 6 years. So in 2022, not very far away from where I am actually now, Svante Pääbo, the Swedish biogeneticist, received the Nobel Prize. And he received the Nobel Prize for medicine, and we might want to talk about what that means. He received his prize for work on ancient DNA. That is DNA derived from samples found in archaeological sites. And his particular expertise was work on a site in Siberia called in a place called Denisova, and on a set of remains that have become known as Homo Denisova. And he also works very closely on Neanderthals. And in his Nobel acceptance speech, Pääbo said this: "The information extracted from ancient genomes contributes to our understanding of the evolution and history of humans and many other organisms." So my project up to this point has kind of responded to what Pääbo is talking about here. The idea that information extracted from ancient remains, and in fact contemporary remains, and turned into genetic data can change our understanding of history. As Stefan said, I published a book relatively recently about this particular thing. And so these are some of the questions that have kind of come to my mind as a consequence of this. I wonder what the implications of such expanded genetic knowledge are for us as historians, if we are historians, how this might change our experience of the past, or even to change what we think the past is. And this is a very good example. I think Pääbo's Nobel Prize is a very good example. Until Pääbo's work, we kind of had a bit of a cliche that Neanderthals were stupid, that Neanderthals were an inferior type of human. It becomes very clear through his work and that of others that this is not true. So actually this genetic data has allowed us to understand this relative of Homo sapien in much more detail, and therefore rethink our own understanding of the human in history. I also wonder what this type of knowledge does for historical knowledge. Is this data? Is this evidence? How do we understand and read it? It's highly specialized, the information that is generated here. How is this able to be kind of used by us in the humanities or in libraries or other spaces? And why is this happening now? Why did Pääbo, who had been working on this material since 2008 or thereabouts, receive the Nobel Prize in 22? Why is there so much information being spread around and shared about this data at this point? I guess my basic question was, are geneticists historians? This is the historic reading room in Duke Humphrey Reading Room in the University of Oxford in the Bodleian Library. And this is the Ancient DNA Lab in Leipzig, where Pääbo did most of his work that I visited about a month ago. And my question is, what's the similarity between work being done in each place? Can we basically make the call that geneticists definitely are historians, or historians might be geneticists? And I think we can. My claim really is that the work that's being done by in these labs is historical, and it changes what we might think of and conceptualize of as history. And I'm going to try and demonstrate that by thinking specifically about manuscripts and books, and those that we have in the Rylands. Because in the last sort of, I guess, 10 years, we've seen the rise of what is called biocodicology. Biocodicology is a means of looking at texts in an archive and analyzing them from the point of view of the genetic data that they hold. And there are 3 ways we might do this. The first is DNA analysis, which is relatively straightforward. We would take a sample from a piece from parchment, from vellum, And this allows us to identify, to a pretty good degree, the type of breed, the type of species, the type of animal used to make the material. This has been most spectacularly demonstrated in the case of the Dead Sea Scrolls, where work has been done to demonstrate that the connections between certain of those scrolls because of the animals that were used to create the skins that they were written on. So DNA analysis is one way we can do this. This is relatively straightforward. There are kind of problems here. For instance, in DNA analysis, the relationship between sheep and goats is quite difficult to differentiate. So actually, whether we know about which species is one thing, but actually whether we can see things in such detail is another. We can also do what's known as protein analysis, and that's what this person is doing in this slide. They are essentially withdrawing some of the protein from the manuscript, generally collagen, and you can do some work on that, and you work on the collagen, identify the type of tissue that is being used for the manuscript, and speculate about environment. And both these types of analysis and these ways in allow us to really rethink the idea of what a text is and how we understand it and comprehend it. And the third type of biocodological work is microbiome analysis. That is using chemical analysis to look at the genetics of the microbes that are associated in the object itself. So it's called biocodicology because, like codicology, it seeks to understand the relationship between texts and essentially uses biological intervention as a means for understanding that. The person who's written the most about this is a woman called Sarah Fiddiman, and this is what she says in a text written for us in about 4 years ago. She says, aside from the text, the physical parchment object holds vast quantities of biological information that, though in many cases is invisible to the naked eye, can be used to provide a deeper level of understanding about book production, livestock economies, handling, conservation, and the historic use of the object. And that's what I'm really fascinated about with this. She's not interested in what's in the book. She's not interested in the writing. She's not interested in the importance of the book even. She's not interested in why this book might be considered valuable, significant, etc., etc. This is a means of approaching an archive which really sees the archive as a repository of biological data. And so therefore, as something which can be read in that way and what can be used to answer certain questions which are possibly not anything to do with the reading of the text or the linking of the text to other texts, the language of the text, what the text looks like even. We're not interested in any way in this kind of analysis, really, with what the book might be or what the manuscript might mean. I'm interested in this. I mean, on the side of the screen, you can see some of the images from Fiddiman's work here. And what I'm interested in with this is that this is kind of reducing a text to something which can enable us to start to map in various different ways. So it can give us all this kind of really interesting new information and answer questions that we maybe hadn't even thought we could ask about texts. I quite like the irony of the fact that basically to extract some data from the manuscript, you use an eraser. So as I talked about with the proteomic analysis to take collagen, to extract collagen, you essentially use a PVC eraser to withdraw that. And it seems that actually cleans the manuscript rather than anything else. So there's some irony in erasing a manuscript to find information. And the last thing here on this screen is one of my Project Manchester's DNA that Stefan talked about, but also the image is of the printed genome, which is one of only 3 printed genomes in the world, in Europe, sorry, one of which is in Manchester, and it's in the MRI library on Oxford Road. The reason I have this here is that I love the kind of feedback that's going on here between the book becoming something which enables us to understand life, and then this printed genome being the book of life in a book itself. It's really fascinating, and I would really recommend going and having a look at it. It's in their open library. It's just on Oxford Road, and it's huge, as you would imagine. But it's pretty much unique. Well, pretty much, sorry, rubbish. It's not unique. There are 3 of them, but it's the only one in the north, and there's only 3 in Europe. So it's worth going and have a look at. Obviously, because it's the printed genome, it's GATC of all of the human genome, it's an incredibly small print, and you have to have a magnifying glass to look over it. Here are actually, so I talked about proteomic analysis. Here are some very up-to-date and new proteomic analyses that were generated for for Ingrid Rembold, who's a colleague from history. She did some work with Chetham's library around the medieval quarter festival that happened in September, and she sent some samples from their books to the lab in York that does this kind of work, and this is what was generated. So she could demonstrate using this information that these are the types of animals that these books were derived from, these samples are derived from. And what I'm fascinated about here is not necessarily the result sheep, cattle. Sorry. What I'm interested in is that this is a way of thinking about books which introduces a huge amount of data, and we need to find ways of thinking about and reading that data. The other way we can think about the manuscript from this point of view is microbiome analysis, as I said. And this is some work that was done on 1630s death registers in Milan by a team led by Alfonsina D'Amato. And her team did some work where they analyzed the microbiome of these pieces of work. They looked at the genetic makeup of the things in it. And this is what they find. Family proteins, mouse keratin, human tissue protein, mouse and rat protein, plant protein, mainly to do with food. So the first thing is to say about this, I think, is yuck. But the other is to recognize that actually the archives that we use and the books and manuscripts that we use are teeming with life. And the analysis of the microbiome of the archive leads us to start thinking about the ecology of the archive. And in the words of Joshua Calhoun, we need to maybe start thinking about a book's ecological history. D'Amato and her team said this about the book: "We decided to explore these documents to extract information not accessible to librarians, historians, and archivists, but who typically try to decode the writing, but have no idea of the wealth of information stored on the support." And I'm very keen on accentuating the binary difference that's being presented to us between what librarians and historians do, that is decode and read, and what the scientific team do, which is to understand the storage system that's being presented to us here. That we can use this to find out data about mouse and rat proteins from the 1630s. And that's what she's kind of keen on. And that's what Calhoun means, I think, by the idea of a book's ecological history. And I'm interested in the idea of the book becoming a site. On the side of the slide here is quite an unpleasant slide. It says, "Do not feed the fish" in Swedish. At the University of Gothenburg, they have a problem with silverfish that are coming into their library and eating the books. And they're being encouraged in by people eating in the library and getting crumbs on the books. So does this lead to new knowledge or different types of knowledge? I'm kind of interested in this question. Does this allow us to say something new, or is it just a new way of thinking? On the left-hand side, you can see some analysis of a nun's teeth. They found in the tatar of her teeth, they found lapis lazuli, which suggested that she was involved in the production of illuminated manuscripts. That's new knowledge. We suspected this, but we could not prove it. And on the other side of the slide, the Te Tiriti o Watangi, which is the Treaty of Watanga in Aotearoa on New Zealand, very important document for Maori and indigenous land rights in New Zealand. It's on two pieces of vellum, and this analysis has demonstrated they're from the same type of sheep, same type of ewe, and there's actually a genetic relationship between the two. So that basically is they were able to say that this is genuine, and the two pieces are genuine. So here are my questions. What can the DNA of the materials of the library tell us about collections, about curation, and about knowledge itself? And how does this challenge and change the role of the librarian, the archivist, the researcher, and the curator? And at this point, I'm just going to I'm going to stop sharing my screen and hand over to Jeremy to talk a little bit about this piece.
Hi, everyone. Yes, so we have a piece of papyrus in front of us here. It's a fragment. It was found in Egypt, and it was acquired by the library in 1917. It was bought from an antiquities dealer, so we don't know where it comes from in Egypt, which is a particular problem, but it is what it is. As I mentioned, it's a piece of papyrus, which is a plant that grew in the Nile. I think nowadays there's maybe not so much papyrus reeds growing in the Nile anymore. I think I read somewhere that the largest source of papyri nowadays comes from Florida, but back in 2,000 years ago, it was the Nile. As far as the contents of the text go, it's quite interesting. We have some peculiar handwriting on it, which scholars have really puzzled over, not quite sure what to make of it. So there's been a range of dates suggested for this text based on the style of the handwriting. Some have suggested 3rd century. You can see from the style of the language is Greek. You can see from the style that the letters are quite narrow and long. And so some scholars have suggested that that style comes from the 3rd century. The problem is we have this, we have alphas here that are really not like anything else we've seen from that century, and are actually more indicative of the style of writing someone might include for an inscription on stone. So some have suggested perhaps this text is a model for an inscription. So we're not really quite sure what to make of it. The other interesting thing I'd like to mention about this text is the content itself. Did I mention it's a prayer text? This is a prayer text. It seems to be a private copy because we don't have any indications that it was used in a public setting or in a liturgical setting. And along with the suggestion of a private setting, I'm not sure if you can see it, but there's a crease here, there's a fold here, and there's a fold here. So it looks like maybe somebody had a private copy of a prayer and folded it up and put it in their back pocket at some point. But anyways, on to the content, which is quite interesting. We see a word over here, theoteka, which in Greek means bearer of God. And so some have suggested if this text does indeed come from the 3rd century, that this might be the earliest example we have of a prayer text that refers to Mary as the mother of God, which is an very interesting suggestion. I think the earliest other example we have is from the 5th century. We don't even have prayers that include Mary without this title in the 3rd century. So this would be quite extraordinary if this is correct. But anyways, yeah, it's a prayer to Mary. It's a petition in which the one praying asks Mary for protection from danger. Much like in the Lord's Prayer, for example, when there's the petition for Jesus to protect the prayer from evil or danger. So aside from this very early date, others have come along and suggested that perhaps it's from the 8th century. There are some indications within the style of Greek itself. People have found more recently some parallels between some 8th century texts, so it could possibly come from there as well. So it's a huge range. Lots of problems with it that we haven't yet been able to sort out properly. Is there anything else you'd like me to say about this text, Jerome?
That's fabulous, Jeremy. Thank you so much. Can we just stay here just for a second on the image itself? And really, so the approaches that I've been describing, Jeremy talked wonderfully about this piece, but also about what we know about it, but also what we don't know about it. And quite a lot of those unknowns can possibly, I'm not saying they will be for this, possibly start to be addressed by some of this type of work. That's the promise of the work anyway. Whether we believe it or not is a different matter. And so it's interesting to think about what questions we might want to ask about a text that may be answered by the intervention of particular scientific method. I'm thinking in this case about thinking about genetics to do with the plant matter that this was made of, although that would probably be quite difficult to do. But actually, we thought that would be difficult to do 10 years ago with animals, and that is now relatively straightforward. So the kind of speed with which stuff is changing is important for us to recognize. Moreover, we might think about things like ink, and we might think about the way in which we could, if we could take gain information from what the DNA analysis of how the ink has been made or what the ink is made of. There are ways into this. The other kind of sort of approach that I've talked about briefly is about this microbiome approach, which would be to ignore what's on this entirely, to not care about the writing on it whatsoever, but to think about this as a repository of biological information. And what does it have? What is residual in the microbiome of this piece? And therefore, what can that tell us about particular aspects of historical experience? So I think that this is a really beautiful piece of text. It's a very beautiful object, and it really makes us think about the materiality of the text in the archive. But I think that what the biocodicological work can allow us to do is to really resituate even such a beautiful and well attested and well discussed piece as this in slightly different ways. What I'm also interested in is whether the biocodicological work can urge us to kind of reconsider what this text is and why we care. Essentially, why we're bothered about this text. I said I'd talk a little bit about the future, and so I'm going to go back to sharing my slides and talk to you about the future just briefly before we come to questions. So one of the kind of amazing things that I've discovered researching DNA is a move which has been gathering some pace over the last 4 or 5 years to use genetic material as storage. Basically, what you do is you synthesize DNA and you use it as storage. So on the right-hand side of the slide, as you look at it, you have the electronic data, ones and zeros, you binary it. You then turn that, you encode that into the basis of DNA text, GTACT. You synthesize that, you store it, you retrieve it, you sequence it, you read it, which all sounds very simple. It's not at all, but the reasons for this are multiple. One is that you can use these to store a lot of the data on very small, in very small spaces. You could probably get most of what's in the British Library in something about the size of a table. And the two other kind of reasons that people are interested in this is that this cuts down massively on the energy associated with storage, particularly of electronic data. And it also, the theory goes, because DNA is so foundational to the way that we understand the human, we will always be interested in DNA, so this will always be readable. So the DNA Storage Alliance group, for instance, wrote about this, saying they wanted to create and promote an interoperable storage ecosystem based on DNA as a data storage medium. And one of their big guys, Nicholas Guys, talked about storing an exabyte of data, which is pretty huge, in a volume the size of a storage cube. You can kind of see the obvious interest in this. And indeed, there is quite a lot of money in this now. The market for DNA storage is going to be around about 1.5 billion next year. And various of these companies that are working in this have been given quite a lot of venture capital money. But it's very much happening now. And we're starting to think about DNA as a storage medium. Here is Guys himself working on the nanochip, which would write the DNA, which would then be used to encode, which would synthesize the DNA, write it, used to be to encode all the data. And that is the nanochip. And I just put this in contrast with Ramelli's reading wheel to sort of point out that new technologies of reading and writing are not new to us, but actually we might want to sort of think quite carefully about the consequences of this. And whilst some of you may be looking at Ramelli and thinking no one reads like that, I put it to you that next time you open up your phone screen and scroll through, you'll be doing a very similar thing to him. So just to finish with, this is what the BBC said about using storage like this. On the right-hand side here, you have an image that was sequenced into DNA, a GIF that was sequenced into DNA. And Paul Rincon, reporting on this in 2021 for the BBC, said, using life's preferred storage medium to back up our precious data would allow vast amounts of information to be archived in tiny molecules, which kind of brings me back to the start. Essentially, what's happening here is that the book of life, that is genetics and DNA, is being turned into an actual book. It's being turned into, we're understanding now the life of the book. Rincon's idea that this is life's preferred storage medium really resonates with me. It feels like there's some sense that we are kind of moving toward a biomolecular solution for storage and archiving, which has a kind of an aesthetic and imaginative neatness, which feels a little bit unlikely. Okay, so I'm going to stop sharing my slides now because we've come to the end, and it's now over to you, everyone, for your questions and thoughts. Stefan, hi.
Thank you. Thank you so much, Jerome. Thank you so much, Jeremy. This was a really fantastic and inspiring paper on the materiality of text that really invites us to think about innovative scientific materials approaches to textual artifacts, which is very much at the core of things happening here at the John Rylands Research Institute and Library. So thank you very much. This is the moment where I'm opening the floor for questions. So everybody out there, if you have some questions, please make use of the Q&A function. I'm going to forward and share these questions then with you. Whilst I'm waiting that some more questions arrive, I wanted to ask a question. I mean, Jerome, this is absolutely fantastic to think about the biomolecular approaches and the kind of interdisciplinary potential of such approaches to establish new connections between different fields of knowledge, disciplines, in the creation of new knowledge. My question is about the interpretation of the scientific data that is generated. So is that an area where the humanities expertise, historical, literary, curatorial expertise comes to the fore again? Is that something you would say?
Yeah, so I'm trying to figure out what this means for us in the humanities, to be honest. And that is not just, this is a sort of case study on something which I think brings this analysis into the heart of what it is to be a humanities scholar, because it's a library, it's an archive, this is what we do, right? But actually, this is also appertaining to lots of the archaeological genetic data that's being created as well. There's a huge archive of information out there now, which is being added to every day by a genetic analysis across the world, and we can't get at it. We can't use it, really. We don't have the training, we don't have the understanding, we don't know what questions to use. And I'm kind of, first off, when I asked this question, our geneticist historians, the reason I asked that question was I wanted to conceive that if they were historians, that then what would they need? How would we need to understand what they did within an historical context? You know, how they dealt with narrative or evidence or ethics, you know, things that were major issues and questions for within the historical kind of profession and approach. I'm kind of changing my mind about that now. I think they are definitely historians, but what I'm interested in is, well, what does this mean for the humanities more generally? What is the future of the humanities if we have two things? One, we have an archive which is biomolecularly understood, and we have a human who is biomolecularly understood as well. If we have those two things, where do we go in the future, in the next years? And on the one hand, you can say, well, we can do what we've always done in the humanities, which is kind of sort of critique from the sidelines. And that's fair enough, because we should be doing that kind of work, which is kind of critical of this. Or we should find ways that we can engage with and analyze. And I don't know what happens with that, because it's very difficult. And what tends to happen in projects like this is you have what I would call multidisciplinary work rather than interdisciplinary work. So you have a collaboration in which, you know, someone does this, someone does this, someone does this, and you know, someone brings it together. But I don't know, if I talk to my colleagues who work with people in evolutionary biology, I'm not sure that their contribution is changing the evolutionary biology approach. You know, it might change what we do, but it's not changing, it's not pushing back. Not there has to, but there's, in terms of a true collaboration. So in terms of the data, yeah, I think we are definitely here to be working on the things that we've always done, which is interpreting and thinking carefully and thinking about ethics and thinking about longer histories and all this stuff. Absolutely. Whether we can do that or not is a different matter entirely. So there's a fascinating set of papers around this from someone called Monica Green, who works on pathogens, and she's an historian, so she works on the plague basically. And she sort of says, yeah, this is amazing, we can ask all these new questions, fabulous. But the problem is that if you don't have the training, you may not be able to conceive of what the questions might be. And equally, those without the training who are doing this work are kind of asking the questions without us. So in some ways, they don't care.
There have been a few more questions that have come in the meantime. One of the questions is about the perception, the public perception of these intersections between genetics and history. So the question is there a perception that genetic tests provide an objective, infallible kind of truth, supposedly missing from historical research? How is that something that we engage with?
Well, yeah, absolutely. So the still continuing popular understanding of genetic data is that it sort of has a truth associated with it, and that derives from both science, the metaphors used in scientific journalism, but also from things like paternity tests and DNA testing for family history and things like that, which still have a kind of resonance of truth around them. So yeah, I think the public perception is still, this is real. Even though if you talk to anyone who works in it, they will say, we're just telling you this, here's our ethics, here's our caution. I don't think anyone is making truth claim. That said, quite a lot of the major claims and major kind of interventions of ancient DNA work over the last 10 years have been quite big. And they have been saying very big things. And partly that's because they've been making noise. They've been wanting to generate noise. And if you read some of the papers, early papers particularly, they do say, this is changing the way we understand the past, full stop. And, you need to be a bit careful of that kind of bombast. But yeah, certainly, I think that the public perception of truth is important. Also, the public perception of kind of DNA as something which shows us something essentially about ourselves, which we cannot escape. And I think that's kind of crucial as well. And I quite like one of the things about the fragment that I think is really fascinating, they've just started extracting DNA from things. So they've extracted it from, for instance, a bone pendant. So someone wore this pendant and sweated. And so the pendant has their DNA in it. So they've taken DNA from that. So that we may not be able to say anything about the parchment. We may be able to say something about the people who read the parchment. And that's kind of astonishing that we can do that. And it's a bit like, it's kind of basically CSI prehistory. Basically, we know that we can't do anything without DNA traces. Well, actually, that appertains to people thousands of years ago as well. We can start to reconstruct, which is kind of mildly mad, I would say. Sorry. I'm talking too much, Stefan. Sorry, I'm massively overexcited. I'm sorry.
We all are, and that is also evidenced in a number of further questions that have arrived. Okay. There's a question which is for both of you, so maybe Jeremy, as the curator in the room, could start with a response, and then hand over to you, Jerome. Do you foresee any long-term difficulties in archiving data in this way? So could it create barriers to entry for research institutions without access to this technology, for instance? What other kind of difficulties from a curatorial perspective in archiving such data? Jeremy, if you want to start as the curator, otherwise
Well, I don't know, I'm not an expert in this area. Maybe it's best if I just turn this over to Jerome. Of course, institutions that have the money, the technology to do this research will be able to engage in this, creating this new forms of knowledge. So obviously, it will divide, as we move forward into the future, it will divide some institutions from others, depending on the types of questions we want to ask of the material. But of course, I don't foresee long-term difficulties for asking traditional sorts of questions that we have asked in the university.
Jerome, do you want to add to this?
Yeah, I mean, one of the really interesting, I just talked with Professor of Paleogenomics here, and she was basically saying they're just creating so much data they can't keep it anywhere. So the people who are doing this work are just generating so much information. And like I said about the future of the archive, we need to find out ways in which we can keep that stuff. And it can be, as you said with your first question, that it can be then interrogated by people in 20 years time or in 100 years time. It has a kind of, it's not obsolete as information. It's not just about now, but it can have a kind of life. So there's that. The other kind of fascinating answer to this is about who's playing these games right now, who's putting this money in. The venture capital I talked about for the future archiving types of technologies is from the US intelligence service, it's from Microsoft, it's from big players in biotech. So in some ways, this is a very old-fashioned story about archives, which is it's about money and power, and it's about ownership. And basically, the people who can run these systems the best will have all kinds of ability to think about how it relates to quantum to DNA computing, to molecular computing, to infrastructure, to encoding, and all. There's all manner of things that come with this. And the money that's coming in is not coming from humanities institutions, libraries, or organizations. It's coming from states, and it's coming from big biomolecular biotech companies already. So the kind of dystopic version of this is that it will be being developed by people who do not have libraries' best interests at heart. Maybe I'm being a little unfair there, but yeah, that's not that's not what this is about to begin with. It's not about let's do this for the good of all humankind. It's about let's get one over on everyone else. Maybe I'm being cynical.
Thank you very much. I'm also aware of time, so there have been a few questions that all address the multi-species angle of your research and of the research that you have presented. And as I said, being aware of time, I'd like to bundle them and invite you to elaborate a bit more on this, the multi-species angle of the research that you have presented. There is a huge number of people out there who's really excited about this.
Yeah, can you say a little bit more about what you mean by the multi-species?
The interactions between humans and other species that come.
Yeah, absolutely. So I mean, so Josh Calhoun's work I really recommend to anyone. He works on foxing. So he works on the spots that you find in an old book, and which we know a little bit about. We don't know much about. And he basically says we need to think about the non-human actor in the archive. And I think that's fascinating. We need to. I mean, there's a load of approaches in my field called eco-criticism, which are eco-critical approaches to thinking about getting rid of the human. That's also the case in archaeology. We're interested in objects in archaeology which are not necessarily associated with human use. And I think thinking about books as things that don't necessarily, that aren't solely for human use and that have an ecology of their own is fascinating. But yeah, also this work allows us to think about the animals that were involved in production of books, the animals that were killed and used in this way. We can think about production and commodification across Europe, across the world. We can think quite carefully about different types of contribution and how we might understand that. And quite a lot of this work is coming from the point of view of animal studies and coming from the point of view of biological animal studies, people who are interested in this not because of the data that it produces about religion, history, but actually it tells us so much about migration, movement, animal use, husbandry, economies, production, soil, etc., etc.
Thank you very much. As I said beforehand, I'm aware of time. We are almost at the end of our today's session. Thank you very much, Jerome. Thank you very much, Jeremy. This has been a really inspiring, fantastic presentation. And thanks to all of you for your audience questions and comments. And I'd also like to thank our tireless tech team who have been running this for us off camera as well. Our next seminar will take place on the 30th of November, 1 p.m. and is entitled New Light on the Oldest European Printed Book, the first edition of the blog book Apocalypse, Rylands 3103, featuring Dr. Stephen Mossman and Tony Richards. So there's going to be more about cutting-edge approaches to our collections. You can find more details about this on the What's On page on the Rylands website, and you can register from there with the Eventbrite link. And yeah, thank you all for joining us, and we hope we'll see you all again on the 30th of November. Thank you very much.